Good morning students. Today 12th standard biology chapter 6 molecular basis of inheritance and transcription and unit and the gene. What is meant by transcription, a basic idea, and what is meant by gene. Normally we know during a transcription process. What is meant by transcription? So far we are completed DNA, the structure of DNA, the molecules are present in the DNA, a monomer of DNA like a nucleotide and nucleosides, and DNA replication procedures, and various enzymes are involved in the replication procedure, and what is meant by replication, all this done, and the basic idea about a transcription also done. Now what is meant by gene and what is meant by transcription, a basic idea. What is meant by transcription? Transcription is nothing but the formation of mRNA, the formation of RNA. Maybe we know that RNA is divided into some types like mRNA, tRNA and rRNA. The three RNAs are present: mRNA, messenger RNA, and tRNA, transfer RNA, and then rRNA, ribosomal RNA. The three RNA present in the cell. So normally we know it is actually a cell, animal cell, which contain nucleus. This is eukaryotic cell, so it is a nucleus present, and chromatin reticulum also present, the nucleolus. In the chromatin reticulum, actually the cells need the production of protein. For example, the cell need actin, a protein, or the cell need a salivary amylase, enzymes. This is actually a proteinases enzyme. Suppose the cell need protein, the cells is actually contain DNA. The DNA only has the information for making the protein. In the DNA only the information are present, which is useful for making the protein. So the information from the DNA is copied into mRNA. Okay, the information from DNA is copied into mRNA. Listen. So DNA like information, it is copied into mRNA. Then mRNA enter into the cytoplasm of the cell with the use of ribosomes. This is actually a ribosome. With the use of ribosomes, with the use of mRNA, then tRNA, then rRNA, lastly the cell produces the protein, a particular protein. So protein production information is present in the DNA. So transcription is nothing but the formation of mRNA, the formation of tRNA, the formation of rRNA, the formation of RNA from the DNA. So from the DNA, the RNA is formed. The process is known as transcription. Okay, in our topic, the functional unit of inheritance. So actually we know the gene is a functional unit of inheritance, the structural and functional unit of inheritance. How the characters are passed from one generation to next generation? Through the gametes. The gamete contain 23 chromosomes in human. Then through the gamete the genes are present. Through the genes only the characters are passed from one generation to the next generation. Okay, so through the genes only the characters are passed from one generation to next generation. So gene is nothing but it is a unit of inheritance, and particularly functional unit of inheritance. Okay, then DNA sequence, the code for tRNA, rRNA, also known as gene. For example, the DNA. Listen the drawing. This is DNA. The DNA, listen. This is the part of DNA I mentioned in another color. So there is a part of DNA which codes for the what? tRNA. DNA only it's code for mRNA or tRNA or maybe rRNA. So part of the DNA which code for RNA, that is known as what? Gene. With the part of the DNA which code for RNA or maybe rRNA, maybe mRNA, or maybe tRNA. The part of the DNA which code for the RNA is known as gene. Listen, the DNA sequence a code for mRNA or tRNA and rRNA. tRNA means transfer RNA, which is useful for the protein synthesis process, which is actually tRNA present in the cytoplasm. And rRNA means ribosomal RNA present inside the ribosomes. Okay. So the DNA sequence, the part of DNA which code for the tRNA or maybe rRNA or maybe what? mRNA, also known for gene. Sir, so one part is coding for mRNA, one part is not coding for anything. So appo adukku enna vechikalam? So up comes some differences between gene. So gene, so far we are gene அப்படினா என்ன பார்த்திருக்கோம்? Gene is a segment of DNA. The segment of DNA which is controlling the what? Characters or controlling the inheritance. It inherit the characters from one generation to next generation. That is known as gene. So some genes which code for mRNA, some genes which code for tRNA, some genes which code for rRNA, some genes which code for protein, some genes does not code for anything. This does not code anything. So adukku gene vekka mudiyuma? Some differences between gene definition. Then the scientists konja discover panni, research panni. So the DNA is a segment of, the gene is a segment of DNA which code for the polypeptide. The segment of DNA which code for the polypeptide. Polypeptide means what? It is a protein. Appa or DNA oda or segment which code for the polypeptide protein. Appa or DNA oda segment protein code pannuchina, the segment is known as what? Cistron or extra name istam. The gene abdingiradhu common name. The part of the DNA which code for the protein also gene. Which part of the gene is a part of the DNA which code for tRNA also gene. The part of the DNA which code for rRNA also a gene. So appo idukku adukku eppadi difference solla mudiyum? So some scientists, so idu adu difference varudhu. So appo some different names kudukka aarambichiralam solittu different names kudukka aarambichittom. So the DNA is a structure present inside the nucleus, we know, in the form of chromatin reticulum. The segment of DNA which code for the polypeptide. Appo or DNA oda segment or particular polypeptide code pannuchu அப்படினு சொன்னோம்னா the segment of DNA is known as cistron. Okay, the segment of DNA which code for the polypeptide is known as cistron. Okay, the next one, in eukaryotes the monocistrons are present. Monocistrons means what is in the eukaryotes? We know it is a higher organism. It contains a well developed nucleus. Nucleus means nuclear membrane, nuclear pores, and double membrane cell organelles are present. These structures are eukaryotes. Give an example of eukaryotes. All in Whitaker's 5 kingdom concept, 11th standard you studied. Monera, Protista, fungi, plantae, animalia. Monera is prokaryotic unicellular, then the Protista is eukaryotic unicellular, and fungi is eukaryotic multicellular, plantae also eukaryotic multicellular, animalia also eukaryotic multicellular. So except Monera, all the 4 categories are eukaryotes. So Protista also eukaryotes. Plantae, all the plants from algae to angiosperm, all the plants are eukaryotes. All the animals from poriferum to mammals, all the animals are eukaryotes, and fungi also eukaryotes, even Protista also eukaryotes. So in eukaryotes the monocistronic genes are present. Monocistrons means what is meant by cistron? I only studied the segment of DNA which code for the polypeptide. So DNA contains various genes, many genes are present in the DNA. A segment of DNA which controls, which code for the polypeptide is known as what? That is gene, and particularly here it is cistron. The segment of DNA which code for the what? Segment of DNA which code for the polypeptide means that is cistron. Okay, in monocistron means in eukaryotes, in all the eukaryotes, Same cistron only particularly code for the particular protein. For example, in human cell, one particular cistron which is important for coding the glycogen. Glycogen is carbohydrate. Adu venda proteins dhaan code pannom. Protein ku examples one collagen, actin, myosin, meromyosin, troponin, albumin, globulin, casein, histone, non-histone, the various proteins present in our cell. So and the proteins la nam oru protein mattum vechikalam. For example, in our molecular basis la irukra histone gra protein vechikalam. So histone is the example for protein. Okay, so the histone gra protein produce பண்றதுக்கு DNA present ஆயிருக்கணும். Then mRNA, tRNA, rRNA help oda the histone protein are produced in the ribosomes. Normally we know in 11th standard ribosomes is a protein factory because proteins are produced in the ribosomes. Seriya? Then eukaryotes for example human cells la or particular DNA segment which is code for the histone protein production அப்படினா the same segments, same arrangement, adenine, thymine, cytosine, guanine, same arrangement in the DNA only, it's code for histone in all the organisms. அப்ப histone ங்கற protein produce பண்ணனும்னா eukaryotic cells ல எல்லா DNA segments ஒரே மாதிரி தான் இருக்கும். So all the eukaryotic cells contain the same type of gene which is coding for the histone ங்கற protein. அப்ப eukaryotes ல எல்லாமே ஒரே மாதிரி genes தான். A particular protein, maybe different genes are present, but in the coding for the particular protein, but histone அப்படின protein வரணும்னா in all the eukaryotic cells contain same type of genes, same arrangement of genes, same nucleotide sequence are present. So அப்ப eukaryotic cells ல எல்லா genesம் particular protein produce பண்ற எல்லா genesம் ஒரே மாதிரி தான் இருக்கும். So that's monocistron. So all the eukaryotic cells, all the genes which code for the protein histone, it is similar. So it is monocistron. Monocistronic unit are present in eukaryotes and polycistronic in prokaryotes. Suppose prokaryotic organisms are pathom monera. Monera la archaebacterium, eubacterium. Eubacterium la true bacterium and mycoplasma and blue green algae. So blue green algae, mycoplasma, bacterium, archaebacterium, then all variety of organisms and then the prokaryotes which is not contain a well developed nucleus and well developed cell organelles also absent. So the polycistronic means ஒரு prokaryotes ஒரு genes present ஆயிருக்குங்க. For example, E. coli bacterium contain a variety of genes which is code for the different proteins, and Vibrio cholerae bacterium which contain a various gene and various protein, and blue green algae Nosta, Ganabina, Acylatoria. So various blue green algae which is producing the various genes or various protein in various cells. So அப்ப prokaryotes cellsல பொறுத்தவரைக்கும் ஒரு புரோட்டீனை ஒரு ஜீன் ப்ரொடியூஸ் பண்ணாது. ஒரு ஆர்கானிசம்ஸில் ஒரு பர்டிகுலர் ஜீன் ப்ரோட்டீனை ப்ரொடியூஸ் பண்ணிச்சுன்னா அனதர் ஒன் ப்ரோகரியாட்ஸில் வேறு டைப் ஆஃப் ஜீன்ஸ் அந்த சேம் வேறு ப்ரோட்டீனை ப்ரொடியூஸ் பண்ணணும், மேபி சேம் ப்ரோட்டீனை ப்ரொடியூஸ் பண்ணலாம். பட் இன் ப்ரோகரியாட்ஸில் டிஃப்ரெண்ட் ப்ரோகரியாட்ஸ் கண்டெயின் டிஃப்ரெண்ட் செட் ஆஃப் ஜீன்ஸ் விச் கோட் ஃபார் த பாலிபெப்டைட். ஓகே, அதான் டிஃப்ரென்ஸ். அப்போ பாலிசிஸ்டான் மீன்ஸ் இன் ப்ரோகரியாட்ஸ், சிஸ்டான் இஸ் நத்திங் பட் இஸ் அ செக்மெண்ட் ஆஃப் டிஎன்ஏ. இப்போ யூகரியாட்ஸில் இருக்கிற செக்மெண்ட் ஆஃப் ஜீன், அந்த ஜீன் பேர் மோனோசிஸ்ட்ரான். ப்ரோகரியாட்ஸில் இருக்கிற ஜீன்ஸ் பேர் வாட்? பாலிசிஸ்ட்ரான். ஓகே, in eukaryotes, eukaryotic genes பார்த்தீங்கன்னா split gene arrangementங்கிறாங்க. So normally gene, what is mean by gene? The segment of DNA. It is here a split gene. Eukaryotic, listen the drawing. Okay, this is DNA. We know the secondary structure of DNA, double helical structure of DNA. That is a helical structure. Secondary structure of DNA, that is actually a B DNA. The next one, So DNA only it is code for the what? mRNA. For example, in eukaryotic transcription process the DNA which is code for the what? Actually DNA வந்து mRNA கோட் பண்ணும். So mRNA கோட் பண்ணும்போது different genes இருக்கும். For example இந்த கலர்ல போட்டுக்கலாம். Yes. Okay. This is actually mRNA. But this is DNA. mRNA. Transcription process. After transcription process, DNA forms mRNA. It's actually mRNA in eukaryotes. Here, this is eukaryotes. In eukaryotic transcription process, DNA forms mRNA. mRNA la pathina some are different colors. Listen. In the color la pathina different. It's not in color. This is controlling the different function. This is actually intron. இது வந்து பார்த்தீங்கன்னா exon. And exon intron idea in the next topic la irukku. Okay, during transcription process DNA forms mRNA. In mRNA formation la the mRNA contain exons and introns. Two segments irukkum. Introns ingiradhu useless, adu edukku use agadu. Only exons only it is important for the coding sequence or which is important for the protein production. So in the transcription of DNA is converted into mRNA. Then after transcription, translation process of the mRNA converted into protein. During transcription process, DNA is converted into what? mRNA, and translation process mRNA is converted into protein. So in protein formation and the mRNA exons and introns are present. Listen here, particularly in the mRNA mattum draw பண்றேன் பாரு. Okay, this is actually what? Exon, and this is actually what? Intron. So intron is nothing but the intervening sequence or in between the exons or introns are present. Introns are useless. Introns edukku use agadu. Then next process la splicing அப்படின ஒரு process or maybe in next topic transcription full teach பண்ணும்போது ஒரு idea கிடைக்கும். So in introns are removed and exons are joined. Here exon, here exon, here exon. In the segment 3 exons are present. So exon is nothing but like contain adenine, uracil, cytosine, guanine, the same nucleotide sequence da exons la present are. This is actually mRNA, messenger RNA. RNA also contains same nucleotide sequence. In the thymine, in the place of thymine, uracil are present. All of the difference. So DNA ko RNA ko enna difference? In the place of thymine in DNA, RNA la uracil are present. Okay, that is exons and introns are present. Exons and introns enna na unala exons only it is coding for the polypeptide chain, but is code for the protein. But introns வந்து எதுக்கு யூஸ் இல்லை. So the introns are removed in the next process in the transcription. But in fully matured mRNA la introns are absent. Introns irukadu. Okay, that is actually transcription. And split gene, introns and exons la pinnadi paakalam. Okay, split gene is nothing but in mRNA some part are segmented, splitted. One is exon, one is intron. One is exon, one is intron. So introns and exons ரெண்டுமே மிக்ஸ் ஆயிருக்கனால that is actually split gene. In eukaryotic transcription la மட்டும்தான் இந்த மாதிரி split genes arrangement are present. But in prokaryotic transcription இந்த split geneங்கிற ஒரு கான்செப்ட் வராது. Okay, so in what is meant by transcription the next explain பண்ணும்போது you get the idea what is transcription, what is meant by introns and what is meant by exon. Okay, so in the topic transcription and the gene is a functional unit of inheritance which is important for coding for tRNA and rRNA and ribosomal RNA. Cistron அப்படினு சொன்னோம்னா segment of DNA which is coding for Polypeptide is a cistron. In monocistron, polycistron, the two variety of cistrons are present. In monocistron, eukaryotic cells are present, and the polycistron is present in prokaryotic cells. Okay. The next one is transcription in prokaryotes and the transcription in the eukaryotes. So next one is transcription. Already we discussed, transcription is nothing but from the DNA mRNA formation. Normally listen, this is actually DNA. Actually double helical structure of bDNA, that is human DNA. From the DNA, mRNA is formed or rRNA is formed or maybe tRNA is formed. So from the DNA, mRNA or RNA, DNA lende RNA form aachu. So the process per what? Transcription. The formation of RNA from DNA is known as transcription. So in transcription, so normally in this topic we read, I have studied, I explained types of RNA and transcription in prokaryotes. Types of RNA means what? We know that RNA is three types: mRNA, messenger RNA, tRNA, transfer RNA, rRNA, ribosomal RNA. So three RNA present in a cell. It is important for all the physiochemical process in the cell. Okay. So mRNA, tRNA, rRNA are three types of RNA present in the cell. In prokaryotes also three RNA present. Eukaryotes, animal cell, plant cell, all cells RNA present. Okay, RNA is present in all the variety of cells. In prokaryotes, eukaryotes, all cells. DNA also present in all the variety of cells except some virus. Virus is a separate category. All prokaryotic and eukaryotic cells, DNA present, RNA present. RNA consists of three types: mRNA, tRNA, rRNA. mRNA is a messaging RNA. For example, in the cells, a new protein needed. Cells go protein venum. So adukku yaar message enga irukku? It is DNA. In the DNA only the message are present. So DNA vella poitu work panna mudiyadhu. So DNA, it is giving information to the mRNA for the particular protein. Then mRNA get information from the DNA, get a message from DNA. That's messenger RNA. The messenger RNA contain a message. What message? For the production of protein. And the message yaar kuduthadu? DNA. DNA gives message to the mRNA. mRNA get message. mRNA type a message irukku. Then mRNA move into the cytoplasm of the cell. Cell oda cytoplasm ku move agum at mRNA. Then mRNA in the cytoplasm with the use of tRNA, transfer RNA, and with the use of rRNA, ribosomal RNA, and the ribosomes in the moon structure oda help oda the mRNA which is producing the particular protein. Number cells la variety of proteins irukku because we are contain animal cells. Animal cells, the cell membrane what? That is protein and lipids. Lipid bilayer and protein. Integral protein also present. So protein is important for the histone formation. DNA formation la histone is protein. Cell membrane la proteins irukum. Our cells produce various enzymes for various biochemical process, metabolic process. Various enzymes allame actually protein. Protein is enzymes. So DNA is important for our body. So DNA is important, protein also important. Mainly protein is important. Protein yaar produce pandra? Ribosomes. Ribosomes la smaller and larger subunits are present. Ribosomes only producing the protein. Okay, and then next translation la nama paakalam. How the proteins are produced in the ribosomes. That is next topic, that is translation. Inga DNA la irundhu eppadi mRNA form aagum? The process is transcription. So the DNA, from the DNA, mRNA formation is known as what? Transcription. In the topic how prokaryotic transcription occurs. In prokaryotes how transcription occurs. Listen. So here in the drawing is a transcription in prokaryotes. As an example, bacterium. So we know the bacterium is actually a prokaryotes. So in prokaryotes how transcription takes place. Okay, how in prokaryotes transcription. This is actually DNA. DNA present in where? In a bacterium. Normally bacterium is a prokaryote, it is not contained in a nucleus. So அப்போ DNA எங்கே present ஆகிருக்கும்? Inside the incipient nucleus or inside the nucleoid region. Normally ஒரு bacterium cell central the nucleoid are present. So nucleoid is nothing but incipient nucleus, undeveloped, well developed nucleus. This is known as nucleoid. In the nucleoid region only the DNA are present. That is actually the bacterium DNA. Okay, the bacterium DNA is normally double helical structure, 3' and opposite end 5' and 3' and opposite end 5', then 5' to opposite end and 3'. So that is actually DNA helix. This is bacteria oda DNA. And what is meant by transcription? From the DNA RNA formation ஆகும். அதான் என்னது? Transcription. So அப்போ DNA present ஆயிருக்கு, that is in the bacterium. And in the transcription நடக்கிறதுக்கு some enzymes are involved. Only one enzymes in the prokaryotes, that is RNA polymerase. DNA dependent RNA polymerase. இப்போ DNA வை depend பண்ணி RNA form ஆகுதா? So அப்போ the enzymes is known as DNA dependent RNA polymerase. Simply we called as RNA polymerase. So அப்போ DNA ல depend பண்ணி RNA form ஆக போகுது. So DNA ல depend பண்ணி RNA form ஆகுது. The enzyme is important for the formation of transcription. Formation of RNA is what? RNA polymerase. Okay, இப்போ DNA வை depend பண்ணி RNA form ஆக போகுது. The enzyme is RNA polymerase. Okay, then RNA polymerase in the transcription process, transcription la ஒரு 3 steps இருக்கு. Initiation and elongation and termination. Transcription contain 3 steps. Initiation, first initiation ஆகிறது, அந்த transcription process start ஆகிறது. Then elongation, the particular process elongate ஆகிறது, run ஆகிறது. Then the termination process, the process is stops. So அப்போ DNA transcription, first step initiation in transcription, second step is elongation, third step is termination. So the transcription is over, that is termination. Okay, in initiation part the DNA la irukku, ipo RNA form ஆகும். So நம்ம படிச்சிட்டோம் the RNA polymerase. For example, RNA polymerase is the structure is nothing but that is enzyme, RNA polymerase enzyme. So அப்ப அந்த RNA polymerase வந்து அந்த DNA ல promoter regions ல உட்கார்ந்து. The RNA polymerase is an enzyme which is present in the cell. It is fixed into the particular region of the DNA. The RNA polymerase DNA oda or particular region la vandu just anga vandu fix ஆயிரும். The enzyme is fixed into the particular region of DNA. And the region peru promoter. Okay, so region which is the RNA polymerase or bind, fix the particular region is known as promoter. Okay, the promoter la actually sigma factor அப்படின ஒரு factors இருக்கு, that is chemical sigma factors. அப்ப RNA polymerase வந்து DNA ல உட்கார்ந்துச்சு and for the initiation purpose sigma factor are present. இப்ப initiation ஆக போகுது, transcription start ஆக போகுது. So the transcription initiate ஆகிறதுக்கு RNA polymerase one enzyme is important and the sigma factor another one factor is important. Okay, so அப்ப RNA polymerase plus sigma factor, இது சென்டர்ல இருக்கிறது sigma factor and RNA polymerase, இந்த ரெண்டும் சேர்ந்து first ஒரு இடத்துல வந்து fix ஆகும். Then after fixing the particular what RNA polymerase DNA வை depend பண்ணி அப்படியே RNA வை form பண்ணிக்கிட்டே வரும் பாருங்க. For example, this is actually RNA polymerase enzyme. The DNA வை depend பண்ணி, DNA ல actually two strands இருக்கும். One is coding strand, another one is non-coding strand. Actually coding strand depend பண்ணாது. From the non-coding strand, for example DNA ரெண்டு strand இருக்கா, so இதுல one strand is non-coding, one strand is coding. In non-coding strand இருக்கு பாத்தியா, non-coding strand depend பண்ணி mRNA form ஆயிட்டே வரும். For example, listen, for example இப்ப non-coding ல adenine, thymine, cytosine, guanine, adenine, thymine, cytosine, guanine அப்படினு present ஆயிருந்துச்சு இந்த DNA ல non-coding strand ல. Then mRNA ல என்ன இருக்கும், adenine அப்படியா, adenine னா opposite ல uracil present ஆயிரும், RNA ல form ஆயிரும். Because thymine is absent in RNA, then adenine na uracil. Thymine அப்படினா adenine, cytosine guanine, guanine cytosine, adenine uracil, thymine, thymine na inga adenine, cytosine guanine, guanine cytosine. So அப்ப DNA வை டிபெண்ட் பண்ணி DNA ல பர்టిక్యులரா நான் கோடிங் ஸ்ட்ராண்ட் ஆஃப் DNA வை டிபெண்ட் பண்ணி அதுக்கு ஜஸ்ட் ஆப்போசிட்டா mRNA ஒன்று ஃபார்ம் ஆயிரும். ஸோ இந்த ஃபார்மேஷனுக்கு இனிஷியேட் பண்ணுறது RNA polymerase 1 enzyme plus initiation factor, that is sigma factor. இப்போ sigma factorம் RNA polymerase ஹெல்ப் பண்ணி the process of transcription initiate. Then initiation ஆனோடனே sigma factors வெளில வந்துடும். Only with the use of RNA polymerase automatically the process is elongate, that is process is elongation. Sigma factor வெளில வந்துருச்சு பாரு. So RNA polymerase is a factor which is important அப்படின்னு சொல்றாங்க. ஓகே, ஸோ அப்போ RNA polymerase தான் டிஎன்ஏவை டிபெண்ட் பண்ணி RNA ஃபார்ம் ஆகிட்டே வருது. ஸோ இது ஃபார்ம் ஆகிட்டே வரும், எலாங்கேஷன் ஆகிட்டே வரும். அது பர்டிகுலர் செக்மெண்ட். ஃபார் எக்ஸாம்பிள் இந்த செக்மெண்ட்ஸ்லேருந்து இந்த செக்மெண்ட் வரல இருக்குன்னு வச்சுக்கலாம். ஸோ திஸ் இஸ் ஆக்சுவலி வாட் ப்ரோகேரியட்ஸ். ப்ரோகேரியட்ஸ்னு வாட் மோனோசிஸ்ட்ரான் ஆர் பாலிசிஸ்ட்ரான்? பாலிசிஸ்ட்ரான். ஓகே, ஸோ மோனோசிஸ்ட்ரான்ஸ் அப்படின்னா சேம் இன் யூகரியாட்ஸ், அண்ட் பாலிசிஸ்ட்ரான் அப்படின்னு முன்னாடி தான் பார்த்தோம். ஓகே, ஸோ இந்த செக்மெண்ட் ஆஃப் டிஎன்ஏ வந்து mRNA காப்பி இருக்க போது ஃபர்ஸ்ட் இனிஷியேஷன் ஃபேக்டர்ஸில் ஃபர்ஸ்ட் RNA பாலிமரேஸும் சிக்மா ஃபேக்டர்ஸும் அட்டாச் ஆயிடுச்சு. தென் த டிஎன்ஏல இருக்கிற நான் கோடிங் ஸ்டாண்டர்ட் டிபெண்ட் பண்ணி mRNA ஃபார்ம் ஆயிடுச்சு. அதை டிபெண்ட் பண்ணி ஜஸ்ட் அதுக்கு ஆப்போசிட்டாக ஃபார்ம் ஆகிட்டே வரும். ஆஃப்டர் ஃபார்மேஷன் இந்த அப்படியே ஃபார்ம் ஆகிட்டே வருது. இது இஸ் எலாங்கேஷன் ஆகிட்டே வருது. லாஸ்ட் இந்த ரீஜன் டெர்மினேஷன். ஸோ அந்த டெர்மினேஷன் ரீஜன் வந்தோடனே இந்த RNA பாலிமரேஸ் மட்டும் தான் இங்கே இருக்குது. இந்த RNA பாலிமரேஸோட ரோ ஃபேக்டர்னு ஒரு ஃபேக்டர் ஆட் ஆனோடனே ஸோ அந்த டெர்மினேஷன் ஆஃப் டிரான்ஸ்கிரிப்ஷன் எண்ட் ஆயிடுச்சு. தென் அது வரல ஃபார்ம் ஆன mRNA வெளில வந்துருச்சு. இதுதான் mRNA. ஸோ வாட் இஸ் இன் பை டிரான்ஸ்கிரிப்ஷன்? த ஃபார்மேஷன் ஆஃப் RNA ஃப்ரம் DNA. இப்போ டிஎன்ஏலேருந்து mRNA ஆர் RNA ஃபார்ம் ஆயிடுச்சா? The formation of RNA is known as what? Transcription. So transcription ல ஒரு 4 steps இருக்கு. Okay, transcription ல ஒரு 4 steps இருக்கு. What is the transcription in prokaryotes? நம்ம லெசன்ல இப்ப பார்த்தது transcription in prokaryotes. Transcription is nothing but from DNA mRNA formation or RNA formation. அதான் transcription. Translation ல only one enzyme is involved, DNA dependent RNA polymerase. அந்த பாலிமரேஸ் வந்து டிஎன்ஏல ஒரு பர்டிகுலர் செக்மெண்ட்ஸில் ஸ்ட்ரக்சரில் ஃபிக்ஸ் ஆகுது. தென் வித் த யூஸ் ஆஃப் சிக்மா ஃபேக்டர் இந்த டிரான்ஸ்கிரிப்ஷன் இனிஷியேட்ஸ். தென் mRNA ஃபார்ம் ஆகிட்டே வருது. த ப்ராசஸ் இஸ் எலாங்கேஷன், அண்ட் டியூரிங் எலாங்கேஷன் டைம் சிக்மா ஃபேக்டர்ஸ் இருக்காது, ஒன்லி RNA பாலிமரேஸ் ஒன்லி. இட் இஸ் இம்பார்ட்டன்ட் ஃபார் த எலாங்கேஷன். ஸோ அப்படியே டெர்மினேஷன் லாஸ்ட் எண்டுக்கு வருது. ஸோ ப்ராசஸ் ஆஃப் டிரான்ஸ்கிரிப்ஷன் ஓவர். Then the RNA polymerase ஓட ரோ ஃபேக்டர்ஸ் அப்படின்னு ஒரு ஃபேக்டர் ஆட் ஆயிரும். Then the polymerase அந்த பர்டிகுலர் டிரான்ஸ்கிரிப்ஷன் ப்ராசஸ் ஸ்டாப்ஸ், அண்ட் அதுக்கப்புறம் ஃபார்ம் ஆகிற RNA இன்சைட் த நியூக்ளியாய்டு வெளில வந்துடும். இந்த இன்சிபியன் நியூக்ளியஸில் வெளில வந்துடும். ஸோ ஃப்ரம் த டிஎன்ஏ mRNA இஸ் ஃபார்ம்ட். ஸோ இந்த ப்ராசஸ் தான் என்னது? தட் இஸ் டிரான்ஸ்கிரிப்ஷன். ஹியர் transcription in prokaryotes. In transcription in prokaryotes தான் sigma factor and rho factor. Sigma factor is useful for initiation and the rho factor which is useful for termination. And எந்த enzymes important for the transcription? DNA dependent RNA polymerase. Mainly RNA polymerase which is useful for the transcription. This is actually transcription in prokaryotes. Your 5 mark question or maybe 3 mark question in your board exams chances are more. So study well in your NCERT book. That is transcription in prokaryotes. What are all the enzymes are involved in transcription? DNA dependent RNA polymerase, only one enzyme. What factor which is initiate? Sigma factor which initiate the transcription. What factor is terminate the process? Rho factor is terminate the process. It takes place in prokaryotic organism. It takes place in which part of the cell? It is a nucleoid, incipient nucleus of the cell. It takes place in bacterium. This is actually transcription in bacteria. Okay.